I'm going to introduce you, don't worry. Okay. Buenas tardes o buenas mañanas, bienvenidos después del descanso. Vamos a esperar, yo creo, 2 minutos que venga más gente, pero realmente quería presentar a una persona muy especial para mí, yo creo muy especial para todas las personas aquí en Latinoamérica que han tenido el placer de conocerla. No solamente es una persona con unos conocimientos científicos y unas contribuciones científicas al estudio de la enfermedad de Huntington muy importantes. Yo creo que por favor, podéis prestar atención, porque si no, no se oye bien. Gracias. La doctora ELENA CATANEO, que viene de Italia, lleva trabajando en el campo de Huntingtons 25-30 años, ha hecho grandes descubrimientos en la enfermedad de Huntington. Hoy va a tratar un tema específico, relativamente nuevo, más en vía terapéutica, pero parte del trabajo que se presentó el profesor Ali tuvo parte de base del trabajo que hizo Elena hace mucho tiempo, donde postuló por primera vez que había la posibilidad de que un componente de la enfermedad fuera una pérdida de función. La doctora Cattaneo, además, es senadora de por vida en Italia por sus contribuciones al impacto de la ciencia en la sociedad. Es un gran honor porque esto, para ser nominado senador de por vida, un presidente actual o pasado tiene que escoger un número limitado de personas, es un cargo vitalicio y es la única científica en el Senado italiano. Además de esto, cuando yo no sabía cómo trabajar en Venezuela, acudí a ella a pedirle consejo, porque es una de las pocas científicas realmente con una pasión de cambio social. Fue ella, en la conversación con ella, que permitió el que contactásemos al Vaticano para hacer la audiencia con el Papa. Si no hubiera sido por ella, no hubiéramos nunca podido tener este evento con el Vaticano. Por lo tanto, para mí, ella es una persona muy especial desde el punto de vista científico, desde el punto de vista humano y desde el punto de vista de ayudar a las comunidades aquí en Latinoamérica y a Factor H. Elena, welcome.
Thanks, Nacho, and thanks to all of you for this beautiful meeting and this invitation. I really want to express my thanks with these words. I want to thank you for reminding us what few people with a great mission can do, and even more, that we had the opportunity to be among them. And I also want to thank you for reminding us that what we study in our laboratories does not necessarily have to be separated from its humanitarian aspect. I want to thank you for reminding us that science in uncovering the facts also helps reduce the stigma and the discrimination that Huntington sufferers have long been subjected to. And of course, I mean, we all had the vision that science is also a method to diffuse human weakness towards our patients. And I think we are here really because we share this vision and we want to continue together. So really, thanks for all of this. I will switch to my scientific presentation, which is going to be very simple. And for those of you who are not scientists, the key message of my talk is the following. I will be talking about cholesterol in the brain, and you should consider that in spite of the fact that brain volume accounts for only 2% of body mass, it contains about 25% of the cholesterol that we have in our body. So this is a cholesterol in our brain. It must be important. And then the second part of the message is that I will summarize the data showing that there is a reduction in cholesterol biosynthesis in the HD brain, and therefore our attempts at treating Huntington disease in mice by restoring cholesterol biosynthesis, by increasing cholesterol level in the brain. So, as I mentioned, cholesterol in the brain is very important. The brain contains approximately 20–25% of the whole body cholesterol. The key thing to keep in mind is that the cholesterol we have in our brain comes from local synthesis, because cholesterol from diet doesn't cross the blood-brain barrier. And the cholesterol within the brain is produced mostly by astrocytes. And the neurons use the cholesterol for making their synapses, for dendrite formation and maintenance. Of course, cholesterol is very important in the formation of lipid rafts, intracellular signaling, neurotransmitter release, axonal guidance. It is a very important physiological molecule that, for the brain. And this also implies that the breakdown of the pathway that produces cholesterol and all its metabolites results in several neurological diseases. So this is in physiology, okay, what cholesterol does in our brain. And then, in this slide, I'm summarizing 20 years of work that have shown work done by my lab and other labs, including Michael Hayden and the French group, that have shown that cholesterol biosynthesis is reduced in the brain of Huntington disease animal models. And we were able to show that the reduction, that this reduction is present, can be measured in 10 different Huntington disease rodent models with 4 different methods for measuring cholesterol, its precursors and catabolites. And that importantly, since cholesterol biosynthesis is repressed in Huntington disease brain, I will show you the 5 different approaches that we have used to try to increase brain cholesterol level in 2 rodent Huntington disease model. And I will show you how this treatment have been effective in restoring, in counteracting abnormality in these 2 mouse models. And I will close simply by summarizing the number of animals that we have tested in our trial. So it is almost 400 animals in the last 9 years that have been treated with one of these 5 different strategies. And of course, at the end of the treatment or during the treatment, we were performing a number of behavioral assays. And just to give you an example, we have administered about 1,000 cognitive, novel object recognition tests, so cognitive tests, to these animals, and to show, looking for results, and I will show you the results in a second. This is to say that, this story, seems quite strong. Okay, so this is a cholesterol biosynthesis pathway, and I should say that everything started 20 years ago with this paper published in Human Molecular Genetics. This was the initial study that showed that multiple genes involved in the cholesterol biosynthesis pathway are severely repressed at the transcriptional level in an immortalized rodent-derived striatal cell line shortly after expression of mutant huntingtin. So we were turning on mutant huntingtin, and within minutes, the whole pathway, the level of the messenger RNA of these genes in green that are important, these enzymes, you see, for in this pathway, the transcription of these genes within minutes were severely repressed in the presence of mutant huntingtin, suggesting that this was one of the probably first effects of the mutant protein, at least in cells in vitro. And many of these genes, if not all of them, are actually regulated by this transcription factor. The transcription of this gene is regulated by this transcription factor, SRABP2, and we were also able to show that, in fact, mutant huntingtin interferes with the capability of this transcription factor to translocate into the nucleus, bind to the promoter regions of these different genes, and turn on cholesterol biosynthesis. So this was the first study, and this was in vitro. And since we are, and we sort of an artifact due to the fact that the cells are placed in vitro. So the next thing that we did, we moved immediately to in vivo. And we started looking at the messenger RNA of the same cholesterogenic genes in the brain of Huntington disease mice, and we started with R62, and you see here that also in vivo, in the brain of these mice, we see a reduction in the messenger RNA level for several of these enzymes of the cholesterogenic pathways. And of course, I mean, this was messenger RNA, of course it was PCR, but very soon, I mean, we added many other methods because, of course, these pathways highly investigated, for example, we were able to show that cholesterol biosynthesis in vivo is reduced in the brain of the mice by administering deuterated water, so that you can measure the amount of newly synthesized cholesterol in the adult brain. And also by this assay, we were able to show that in the R62 mice, there is a reduction in cholesterol biosynthesis. But you can do a lot of measurements by using mass spectrometry, and you can measure basically any of these metabolite or catabolite. And in all cases, we were able to detect a repression, a reduced level of these cholesterol metabolites and of cholesterol itself. And this is just a summary, an example of one of these data, mass spectrometry data. And you see here, for example, the level of latosterol in the allelic series. And you see, this is striatum, this is cortex, and you see it goes down. And basically, you can imagine that these metabolites have been measured by mass spectrometry in all these HD mouse models. And in all cases, we were able to detect a reduction in the level of these metabolites and also of these important catabolites, 24-OH cholesterol, which is the only catabolite of the pathway that leaves the brain and can be detected in the blood. And indeed, some groups, including ourselves, are looking forward to the hypothesis of using this as a biomarker of Huntington disease progression. So the data are pretty strong, suggesting that there is a reduction in cholesterol biosynthesis. What are the consequences? Well, on the basis of what I told you at the very beginning, you can imagine that there are very detrimental consequences for brain function due to cholesterol loss. And here are some mechanisms that could underlie brain dysfunction, and in particular, poor cognition in the absence, when you have a reduction in cholesterol level. By the way, aging is also characterized by reduced cholesterol level. In the presence of lower level of cholesterol, the synapses will no longer work properly, lipid raft will be affected, and therefore intercellular signaling. But also at the level of the post-synapses, there are a number of dysfunction that can be measured, like the mobility of the AMPA receptor is affected by cholesterol loss. And last year, a paper in Cell has shown that the TrkB receptor, so the BDNF receptor that promotes neuronal plasticity, has a cholesterol sensing function that mediates synaptic effects of cholesterol. So anyway, this is again to say that cholesterol is important for the brain, and there is strong evidence demonstrating that cholesterol biosynthesis is reduced in the brain of HD rodents. Okay, this is the story. I mean, what can we do? I mean, to try and restore cholesterol biosynthesis and the level of the different metabolites, cholesterol metabolites. These are our strategies to replenish cholesterol in the HD brain, and I will go briefly through them. Let's say our main approach for the moment is to try to supply exogenous cholesterol directly to the brain. How can we do that? The first attempt was published in 2015, and we were delivering exogenous cholesterol to the Huntington disease brain through polymeric nanoparticles made of PLGA, which is a polymer approved by the FDA, loaded with cholesterol. So these are cholesterol-loaded nanoparticles. The nanoparticles are injected intraperitoneally, and because of the presence of this G7 peptide, they can go through the blood-brain barrier, and you can detect them in the brain. And we were able to show that, yes, the nanoparticles injected peripherally were reaching the brain, and in this case, I mean, after the delivery of cholesterol with this first-generation nanoparticle, we were able to show that we could prevent, reverse cognitive decline, and also rescue synaptic activity, while we did not find any rescue of the motor defects. And we calculated that the amount of cholesterol that we were providing through this strategy overall, I mean, in the total brain, with the kind of administration that we were doing, was 20 micrograms. Okay, so some benefits. We then did another study in which we used osmotic mini pumps, and this was published in 2020. This was a dose escalation study, because we wanted to know whether there was a dose of cholesterol that could rescue both cognitive and motor dysfunction. And mini pumps are devices that can be loaded with the desired amount of drug, in this case, of course, cholesterol. They can be attached to a catheter to infuse the drug directly in the brain region of interest. And we tested 3 different doses. Again, I mean, you see here the 20 microgram, the one we used for the first generation nanoparticle, 250, and 500 microgram, which is the highest dose. And with this strategy, we found that all 3 doses were equally able to prevent cognitive decline, while the effect on motor performance was seen only at the high dose, and the high dose was also able to recover synaptic transmission and also morphology, and also to clear or reduce the presence of mutant huntingtin aggregates. Then, I mean, we use a third approach. I told you about SRABP2 being the pivotal transcription factor in cholesterol biosynthesis. So when cells need to make new cholesterol, SRABP2 translocates into the nucleus. It binds to the promoter region of those genes of the cholesterol pathways and stimulates their transcription. And I told you that in Huntington disease, SRABP2, because of the presence of mutant huntingtin, this transcription factor can no longer translocate into the nucleus. So here, we try to force the expression of the active form of SRABP2 with an adenovirus by expressing this transcription factor specifically in the astrocytes, which are the cells that make normally the cholesterol. And we found that also in this case, even though, I mean, this was a unilateral injection, also the mini pumps were unilaterally implanted, we were able, I mean, to rescue, to reduce the number size of mutant huntingtin aggregates, to restore synaptic parameters, and we were able to detect beneficial effects on the motor and cognitive deficits that characterize R62 mice. In parallel, we are working on route of administration, and we are also trying to see whether instead of using nanoparticles, we could use liposomes to deliver the cholesterol intranasally. And this is what we have published something, new work is ongoing, but also in this case, I mean, we can detect the liposomes, the increasing cholesterol, because it is labeled with D6, and also this is very promising. But I want to spend the last part of my talk, I mean, showing you the latest piece of work, the last piece of work. This is unpublished. And so we kept working in collaboration with a group in Modena and Reggio Emilia in Italy to develop a second generation nanoparticle with an increased cholesterol loading capacity, so that we could encapsulate more cholesterol compared to the first generation nanoparticle that was effective on the cognitive parameters but not on the motor dysfunction. And indeed, I mean, with this new formulation, we could measure much higher level of cholesterol in these nanoparticles. So this is the new formulation. And I also want to tell you that, of course, in all these studies, I mean, we can follow the distribution of the cholesterol and of the nanoparticle after injection, because we have different ways, I mean, to measure where they go and to look at their kinetics. And these are tools that we can use to evaluate the distribution, the localization. For example, you can label your nanoparticle with CY5, which is red. You have a red signal. Then you can label the cholesterol with the body P, which is green. And by looking at the combination of the two signals, the red and the green. The red is a nanoparticle, the green is the cholesterol. After injection, you can also monitor cholesterol release from the nanoparticle by looking at the coefficient of overlap between the red and the green signal. And of course, you can measure the profile of in different tissues by labeling by using D6 cholesterol. And we've done that. I mean, these are common tools we use in our studies that have been able to show that after IEP injection, within a few hours, we can detect the nanoparticles in the brain, very wide distribution in all cell types. And we were also able to show that basically 20 weeks are needed to release all the cholesterol from this second generation nanoparticle, suggesting that the release is progressive and slow, which is okay. And with the D6 strategy, of course, we were able to quantify also the amount of cholesterol and the peak, although for the moment we see the peak here in striatum after 2 weeks, but we are missing some time points because the next one here is 10 weeks, so we're doing more work. And you see that in liver and plasma, of course, I mean, the peak occurs much earlier. So these are good tools, I mean, to follow your work. And the other thing I want to mention is that the concentration of D6 cholesterol measured in plasma is around 4,000 times lower than the concentration of cholesterol present in mouse blood. So I wouldn't say that, okay, we are causing a problem, I mean, by injecting these cholesterol-loaded nanoparticles to the animals. And of course, I mean, you can do all your studies, your injection, and measure safety parameters, and we constantly do that, I mean, in all our studies, and we haven't been able so far to detect any immune response, and there are no gross histopathological signs after, I mean, this delivery. Okay, so this is to say, maybe we should concentrate on this second generation nanoparticles, although I already told you that we are already considering the intranasal, but let's say we have so much experience that we really want to see, I mean, whether by delivering cholesterol long term, and we can do that with the second generation nanoparticle, not only long term, but high level, we want to see whether for how long, I mean, we can produce, I mean, beneficial effects in the animals. So I will show you, and these are the data. I will show you 3 trials. The first trial, and here we are switching, we are no longer using R62, we are using the Q175 mice, injected IP with cholesterol-loaded second generation nanoparticles. And the treatment, as we established, I mean, years ago in the R62, the treatment is 2 IP injection of the cholesterol-loaded nanoparticle per week for 5 weeks. So it is a total of 10 injections. And in the Q175, the injection were done between week 5 and week 9, so we're in the pre-symptomatic phase, and we know that it takes about 20 weeks for the cholesterol to be released from the nanoparticles. And then we follow the mice. What are the results? Well, this is the results of this trial, the pre-symptomatic treatment. And we assessed the cognitive performance in these mice by using, once again, the novel object recognition test, which measures recognition memory and is based on the rodent's natural propensity to explore a novel object. And you see that while the wild type, in white, the wild type mice explore a novel object that you put in a cage, the HD mice are not interested in novelty, so they do not explore the object. But in the HD mice treated with cholesterol, basically you restore completely this cognitive parameter at 20 weeks, at 29 weeks, and then you lose the effect. And this, I would say, would be expected. I mean, if you consider that this was an early treatment and that efficacy, or let's say that the cholesterol is released and then is probably degraded within 20 weeks. And if you look at the paw clasping behavior, you look, for example, on disease progression. So here, again, I mean, we know that the Q175 show clasping activity as early as 20 weeks of age, and we see no rescue after cholesterol treatment at this age, but we do see a rescue at 29 weeks, which is then lost later on. And then grip strength, and this measures neuromuscular strength. And here again, I mean, you had the wild type and the HD mice that show a defect in this parameter at all time points. And you see that the cholesterol treatment can rescue this defect at 20 weeks, at 29 weeks, and then it starts decreasing efficacy at 35, and then is lost because there is no more cholesterol around at 45 weeks. And if we measure, if we analyze the animal through this behavioral test, the Rotarod test, at 46 weeks, remember that the treatment was very early on, well, we see, I mean, the expected reduced Rotarod latency to fall in the HD mice and no efficacy of the treatment. Okay, this was the first trial. You give cholesterol pre-symptomatically, and you are, and it shows, I mean, efficacy up to 29 weeks, and for some parameters a little bit also later on. Second trial, trial 3. We did the same experiments, but in this case the injection were done in the symptomatic phase, and all the rest is the same. Again, novel object. You see the wild type, the HD mice, and the mice that were treated at symptomatic stages, and this is the NOR test, the cognitive test, administered at 40 and 49 weeks of age, I mean, in the animals, and you can see that we can rescue the cognitive decline. Po-clasping, I mean, as for the early treatment, 10 weeks administration, because this was just 10 weeks after the last injection, probably is not enough, but we detect some improvement here. Look at the grip strength. We also see, I mean, the recovery. And the rota, if you treat the animals in the symptomatic phase, and of course you measure 20 weeks later when the animals are 50 weeks of age, I mean, you see a beneficial effect also on this parameter. And then the last trial. The last trial in this third experiment, we wanted to see whether we could do 2 cycles of treatment because we wanted to know whether we could, prolong, this beneficial effect over the entire life, or at least 1 year life of the animals. And basically the answer is yes. You see, these are different animals compared to before, and you have full recovery of the NOR test. And look at the po-clasping, ineffective here, but highly effective later on. Also the grip strength and the rota. Not only that, but the double treatment also produces some benefits in this other test that I don't have time to go through. Of course, we're interested at the electrophysiological parameters, and yes, the cholesterol treatments improves some synaptic communication, and also look at the aggregates. Now, the cholesterol wipe out, clear the aggregates, and this is exactly what we saw also in the SRABP2 study and in the mini-pump study. I have 2 more slides and I'm done. So we can say that cholesterol administration is effective pre-symptomatically and symptomatically in these HD mice and produces complete restoration of cognitive and motor defects, and that efficacy, if you do this double treatment, is long-lasting and maintained for the entire duration of the treatment, which is up to 50 weeks tested so far. And here, I like this slide because here we put together all raw data obtained by NOR test. So here we are measuring only the cognitive parameter. In HD mice exposed to any of the 5 different strategies that we tested in our laboratory. And you see them there, the first generation nanoparticle, the osmotic mini pumps, second generation nanoparticles, in R62 mice, in Q175 mice, the SRABP2. And here, these are the results from the NOR test at the last time point measured in any of these treatments. And it looks amazing to me that really, it seems that, by pulling these studies, the distribution of these values from the HD group treated with any of these strategies tell us that regardless of the type and time of administration, delivery system, and mouse line, this value overlap more with the value of the wild type groups than with those of the HD groups. And just to give you some numbers, so here are the 381 mice tested in all these assays, and these are the north, if we consider all time points, and we put them together, all time points, even the intermediate time points, in which we administered this NOR test, we ended up performing more than 1,000 NOR tests in the treated animals. So cholesterol replenishment consistently counteracts cognitive decline in Huntington disease mice in addition to motor recovery. And this is a question for us and for you. I'm looking for help. The question is, how can we turn what it looks like being a very successful treatment in HD mice into something testable in patients? And I really hope we can have your help, expertise, and collaboration. And with this, I'm finished, and thanks a lot for your attention.
Thank you, Elena. ¿Alguna pregunta para Elena? Any questions? Berhard?
So, Elena, thank you very much for a very nice presentation and very compelling data, I have to say. And could you comment whether, to your knowledge, in Huntington's disease, you have observations like in ALS, that levels of cholesterol appears to have an impact in ALS and in terms of survival? And to my knowledge, there are no data in humans that would allow to draw a similar conclusion, but wouldn't it be nice to have to explore experiment of natures in that sense? Yeah.
Yeah. The difficulty is that, I don't know if you are referring to peripheral cholesterol, which is, of course, a different story with respect to central cholesterol.
Absolutely.
The only data, I don't know if, Michael, maybe you want to intervene since you have a lot of experience in cholesterol biosynthesis. The only data, I mean, from patients is, post-mortem data, where you see, reduced messenger RNA for these genes. And, there are studies ongoing that are suggesting that there is a reduction in 24-OH cholesterol, which may reflect a reduced biosynthesis in the HD brain, although, of course, we know that it also reflects neuronal loss. So maybe, I don't know, one could look at statin-treated patients, but I'm not sure that there are really compelling data, but it would be worthwhile, I mean, looking at the different studies and see whether we can come up with some suggestion.
¿Alguna otra pregunta? Any other question? Cristina?
Thank you very much, Elena. I didn't want to make a question. I just want to add two pieces of information regarding this discussion about cholesterol in humans. So we have analyzed the enrolled data regarding the frequency of people that have hypercholesterolemia and people that are on statins. So Swati, that is working at CHI with me, she has been doing this type of work, analyzing the enrolled data on that regard. We don't have yet final results, but we know that hypercholesterolemia in HD is less frequent than in the general population. So there is some kind of interaction between the existence of HD and the presence of the, let's say, hypercholesterolemia as a risk factor for cardiovascular disease. So that's one aspect. The other aspect, because hypercholesterolemia is less frequent, number of people treated with statins is relatively low. And because of the patterns of prescription, the most frequent statin used in enroll is atorvastatin, which does not penetrate the brain. So there is a lot of circular things that make this analysis complex. So that's one. The second piece of information I'd like to say is we have sent all the HD clarity samples, and so that's another example how important enroll and clarity is for clinical research. So we have sent all the clarity samples that are CSF for a full extent cholesterol metabolites analysis. And the data is coming in. So in a couple of months, we will have it.
That's great. Yeah. Thank you.
Welcome, Peg. Peg, o Peginopoulos, es profesora de psiquiatría, pediatría y neurología. Es la presidenta del Departamento de Psiquiatría de los hospitales de la Universidad de Iowa en Iowa City. Es una médica científica que ha trabajado, como he dicho antes, en estudios realmente innovadores y pioneros con poblaciones que tradicionalmente han sido, no diría excluidas, pero sí ignoradas en el tema de la enfermedad de Huntington, como son los casos de Huntingtons juveniles, y ha desarrollado mucho trabajo intentando entender el porqué, la expansión que causa la enfermedad dentro de las personas, digamos, vamos a llamarles normales o no afectadas, es decir, la región que se expande es polimórfica en los humanos. Y se sabe que a través del desarrollo evolucionario de distintas especies, en los humanos hay muchas más repeticiones en las personas no afectadas por la enfermedad que en otras especies. Por lo tanto, se plantea que hay un efecto positivo durante la evolución de tener más número de repeticiones, y ella empezó a estudiar estos aspectos en el contexto del desarrollo del cerebro, y creo que nos va a contar hoy aspectos de ambos proyectos. Peggy.
You guys can hear me? Awesome, yeah. So the first thing I wanted to say, I don't think anyone's mentioned it, but thank you to the translators. I don't know where you're sitting, but I know you're doing a lot of hard work. And hopefully I can speak simply enough. Where are they? Oh, thank you. They've done an awesome job so far, and I'll hopefully be simple enough that it'll make it easier for you. The second thing, of course, thank you, Nacho. So, you know, my work is in children with Huntington's disease. It's not quite up there yet. That's not my first slide. And it's somewhat of a controversial study, and the person who has been my primary supporter for so many years has been Nacho, always there. And then, of course, when I come here, the first thing that I see is all the children. It was really quite striking, and it warmed my heart, and I'm really grateful to all the work that's being done here, and in particular to Factor H and all the work that they've done for the children. So I need my slides up here instead of those slides. So I'm going to start with some take-home points. I'm sorry, what? It's my laptop. I'm not sure what the issue is. But this is what I was worried about. It's always the AV failure. I'm ready to talk.
It's okay, just wait a second. I'm sure we can sort it out.
So yeah, I'll just give an introduction. So I have always wanted to be a singer.